This is Ms. Seidel here. I am going to go through the process of transcription and translation for you. We're going to start with a sequence of DNA and we are going to transcribe it into mRNA, and then from the mRNA I'm going to show you how you can figure out which amino acids those code for. So if we start here, our first DNA sequence is TAC. So we know that the complement in mRNA for a T is A, and then the complement for an A would be T, but since we're dealing with mRNA, we don't have T's, we have U's instead. So in place of the thymine, you have a uracil. And then finally, the complement for C is G. Because remember, A's and T's go together, C's and G's go together, apples in the tree, cars in the garage. So we will go along and do the next triplet here. GAT, the complement would be, see if you can figure it out, CUA. Good. I hope that you guys came up with the same thing. So I am going to pause in between and ask you, and I'd like you to play along just like we're in the classroom and see if you can predict the right answer. So we'll continue along here. GAG, what should that turn into? Hopefully you said CUC. Then we'll head over here. We've got CAT. Yes, it spells cat. That would turn into GUA. And then we have T's here, which should be A's, and then a C. GAC will be CGU. AAG should be UUC, and then finally ACT should be UGA. Okay, so hopefully you got that same sequence of mRNA nucleotides. Now we are going to translate our mRNA into amino acids, and we do that using the codon wheel. There is a fantastic codon wheel on page 303 of your textbook. I also have one that I found online here that we will be using. So this is the same thing that you'll see on page 303 of your textbook. So let's start with our first triplet. Our first triplet here is AUG. So we're going to go over to our wheel. We'll start at the beginning. This wheel will take us from mRNA to amino acids. We start in the middle and work our way out. So we had AUG, and that codes for methionine. And you will notice that methionine is the start codon for all proteins. So that should be the first one that you see. If you're doing a full protein. Sometimes you will have to be transcribing and translating just sections of DNA or a section of a protein, because most proteins are pretty long. You don't typically see one that's as short as this. So let's move on to our next triplet here. We have CUA. So head over to our codon wheel, CUA, and that gives us leucine. Because the name of some of these amino acids is pretty long, we usually abbreviate them with the first three letters. And there are only two amino acids that we can't do that with, and that would be aspartic acid and asparagine. And that's because the first three letters of both of those are the same. So you just figure out a way to differentiate the two, and it should be fine. So our next triplet is CUC. So let's start at the middle, CUC, that one is also leucine. You'll notice that there are several different ways to code for the same amino acid. For some of the amino acids, and then for a few, there's only one way to code for them. All right, GUA. GUA will give us valine. AAC. AAC gives us asparagine. ASP, and I'm going to just add a G. That will let me know that that one is not aspartic acid. Okay, CGU. CGU should take us to arginine. ARG. We've got two more. UUC. UUC is phenylalanine. And then one more, UGA. So that is our sequence of mRNA. UGA brings us to a stop. So that is a stop codon. That doesn't actually, there's no stop amino acid, it's just the stop codon. So what I'd like you guys to do right now is to pause the video, and I'd like you to try to translate this next sequence, sequence 2, into mRNA, and then transcribe that into mRNA and translate it into amino acids. So go ahead and pause the video and do the same thing for both sequence 2 and sequence 3. I'll give you a few seconds, and then once you've done that, go ahead and start the video back up and we will check your answers. So hopefully you were able to do that pretty easily and it was all making sense. You guys can check my work here. It's Friday afternoon and who knows what my brain is doing right now, but what we should have is that sequence 2 is transcribed from DNA to mRNA to get the following. So you can pause the video to check your work and see if you got the same thing. And then I was able to translate those mRNA codons into amino acids and came up with these amino acids. Please let me know if you still have questions, and I hope that was helpful.